Hello, everyone. So, this lecture is going to be all about infectious particles. Infectious particles being something very separate from living microorganisms that are pathogens. So, of course, here we're mostly talking about viruses, but prions and viroids also fall into this category. Please make sure that you review a lot of these concepts in the book because we talked a little bit already about prions. The concept of viroids is covered more in the chapter. This PowerPoint is going to mostly focus on viruses itself. Okay. So until now, we've really focused in on living microorganisms, mostly bacteria. We've talked a little bit about some other different organisms in earlier chapters, but our class has very much centered on bacteria overall. And so, especially in the context of causing disease. That, of course, is what we are often most concerned about, especially in medicine, is the impact that these bacteria have on our bodies. But it's important to note that not all pathogens are alive in that sense. Viruses and prions, these are things that are not actually living things, but they can still cause disease. They can still cause specifically infectious disease. So these are different than, say, if I encountered heavy amounts of lead or something like that, and I accrued lead poisoning. Lead poisoning is itself a disease, but it's not infectious. I can't spread lead poisoning if I have it to somebody else. Instead, these are perpetuating pathogenic particles that can move from one host to another, and so they are infectious. Okay? In many ways, these things behave like living organisms, despite them not actually being alive. So let's actually take a look at that, right? So there are a number of characteristics that living things must have in order to be considered living. Okay? All living things will have all of these characteristics. Okay? Things that are not alive, they will be missing some of these or all of them. So if we look at some of these characteristics that define a living thing, well, a living thing should be able to grow, it should be able to develop, it should contain genetic information, right? It should have DNA or some kind of other source of genetics, not necessarily DNA, but some kind of code that contains all the information for building that organism. They should also be capable of evolving. So this is not something that happens on an individual level. You or I are not evolving, but our lineage undergoes evolution. Our population is evolving. So this is more of a population level thing. They should be able to reproduce, of course, and make more of their species, continue their line, pass on their genetic information. They are made of cells. Very important. They are made of cells. All living things are made of cells. And they also maintain homeostasis. So by that, again, I specifically mean that they're keeping the internal conditions that are necessary for their survival, which is a very difficult and constant job that all living things are engaged in. If a living thing is not engaged in this, again, it is not alive, right? Or at the very least, if it was alive, it will not be for long. So, and then they also are acquiring nutrients from their environment and generating energy. So they are utilizing resources to make energy compounds, things like that. Things that we explored in the chapters about metabolism and things like that. So what about viruses, right? Are they actually organisms? Are they alive? Are they microorganisms? No, they're actually not, because they do have some of these characteristics that are shared among living things, but they're missing a lot of those things. Viruses, they don't really grow or develop. They don't really do that. They also aren't made of cells. They're made of totally different things. They may have a lipid layer that covers them, but it's not a cell. It's something very distinct. They also don't keep homeostasis. They have no mechanism to do that. They don't keep any internal conditions that help keep the virus intact or alive, quote, alive. So that's really important. They also don't eat. They don't make anything, really. Ultimately, viruses make their hosts make everything. So they aren't involved in any part of that process. What characteristics viruses do have that make them very, very much in line with living things, though, is that they do contain genetic information. They do have the ability to evolve. We certainly see that with diseases, viral diseases that change over time. New strains emerge. I mean, for example, we are unfortunately intimately familiar with COVID. That the original strain of COVID was itself a mutation from something that didn't attack humans and it attacked other organisms. It mutated to infect humans and then mutated many other times in order to overcome new host defenses. So the strains that perpetuate today for COVID, they are different from the strains where COVID originated. And that process of mutation and then that mutation becoming the dominant mutation in this population, that is evolution at work. So viruses do evolve. And of course, they reproduce and make more of their species. Sort of. They kind of do that. They themselves do not really do any of this stuff themselves. They make a cell do it for them. And so because of this, because they meet some of the criteria but not all of it, and in fact many of the criteria are missing in viruses, viruses we know are not alive. We don't call them organisms, right? And the same logic goes for prions, by the way. Prions are just misshapen proteins, and so they're also not living organisms. They do perpetuate themselves. They do weird stuff like that, but they're not made of cells. They don't evolve. They don't grow or develop or anything like that. They're just proteins. It's not even. They don't even have genetic information. It is just a protein, and so these things are also not alive, right? We call them infectious particles instead. Infectious particles. And the scale here is very different as well, right? Viruses and prions are very tiny. When we look at viruses, right? So here's a red blood cell up here, and then here's a typical bacterial cell like E. coli, right? And the size difference between a bacterial cell and a human red blood cell is very large as well. But then individual viruses will be much, much smaller than that even. So we're looking at many orders of magnitude difference between like our cells and even a bacterial cell to a viral particle. And then of course, because we know that prions are even smaller than they're just single proteins, they're going to be even smaller than these viruses, right? Okay, let's look actually at viral categorization, how we sort and consider viruses, we categorize them by their anatomy and by their specific features. So a virus can be naked or enveloped. These are the two first main categories that we use to separate out viruses, okay? A naked virus is one that lacks a lipid membrane around it. Okay, and an enveloped one is one that has that lipid membrane. Now, a naked virus, okay, it's going to be composed of Really not much. These are very simplified particles. They contain some kind of genetic information, some nucleic acid. It could be DNA or RNA. It varies depending on what kind of virus we're talking about. And this information, this nucleic acid, is encapsulated in a protective covering called a capsid. A capsid. Okay. This is a protein-based envelope. Basically, it's a protein-based little packet, and it's made of. We call all the little units here that make up the capsid a capsomere. So a capsid is made of many capsomeres, and they can take a lot of different forms. This one looks like a 20-sided die, but there's lots of different shapes that these may take. Very much depends on the virus itself. Viruses come in many different shapes and sizes. But that's the main difference. So the difference, of course, between a naked virus and an enveloped virus is that an enveloped virus is going to have an outer layer on top of this that is made of a lipid membrane, right? A phospholipid bilayer. Okay. Now those enveloped viruses, when they get that lipid membrane, it's very cell-like. It contains. It is a phospholipid bilayer, very much like a cell, but it's notably not a cell, and it's also notably stolen from a cell. So when an enveloped virus is leaving a cell, when it is made inside of a cell and it's packaged up and it's ready to leave, it does so by wrapping itself in the cell's membrane and then pulling bits of it with it, right? This process is called budding, okay? So it's really interesting. You can see that we have these this rather unusual situation where these cells are they're splitting off. Virus material is being packed up in these little buds of cell membrane, and as they leave, they just kind of like encapsulate themselves and split off. So you have here all of this little stuff here. This brown thin layer here is the capsid being formed. It lines the inside of the cell membrane like this, and as it buds, this capsid closes up, and the membrane likewise kind of pinches off, right? And so when it does this is a process that these viruses can use to actually steal the cell membrane itself from a cell, right? And it does this in a way that lets it adorn itself with these unique proteins and glycoproteins. Glycoprotein is just it's a carbohydrate protein mix. It has sugars and protein attached to it, and other features too. These are the things that we call spikes. Okay, so as the virus buds, it takes with it a bunch of these things that were made by the cell as instructed by the viral genetic material. Remember that a virus's job is to get the cell to make all of its parts, right? That includes some of these extra protein pieces like this, right? So, so these spikes, the name should sound familiar to you, because if we relate this back to the subject that, again, we are probably most familiar today, COVID-19, we know that a lot of the vaccines today that we use for COVID-19, they target these specific spike proteins that adorn the virus, right? So that is an important part of what is on these viruses. They serve important functions for the virus, but we can also use them to target the virus with our immune system. Okay, so When we look at viruses, of course, we can separate them out based on them being naked or enveloped, but there are other ways to categorize them too. Some very important ways. So we can further differentiate viruses by their nucleic acid of choice, basically. What their genetic material looks like. Is it DNA? Is it RNA? Because some species will use one and some species will use another. And interestingly, there's more variation here as well, where some viruses, so normally what we encounter is, most living things and most viruses, when they use DNA or RNA, usually DNA is double-stranded and RNA is single-stranded. We can abbreviate that as DS for double-stranded and SS for single-stranded, okay? This is what we typically see for most forms of life, but viruses are rather interesting. They don't necessarily follow these rules. So most of those viruses are going to be either single-stranded RNA or double-stranded DNA. But because viruses can kind of do a little bit of experimenting, they're very strange. We do find some viruses that are actually double-stranded RNA or single-stranded DNA. So it's variable. Now, typically, we do find that most things are either single-stranded RNA or double-stranded DNA, which is more in line with what we have learned so far. Certainly what we see in living things. We pretty much only see this in living things. But viruses are weird, and you can definitely get double-stranded RNA or single-stranded DNA viruses. Okay. Viruses, their propagation is also rather interesting because they are extremely reliant on a host. They require that in order to continue themselves. So these viruses, they need to infect a cell, and they need to utilize the mechanisms of that cell to make more of that virus. Because of that, most viruses tend to be rather host-specific, meaning that they're very, very specialized in the type of cell and the type of species that they parasitize, that they infect. Okay, so some good examples of that would be like HIV. HIV is a type of virus, the human immunodeficiency virus, that only infects humans and certain primates, which are, of course, our close relatives. So it makes sense that that our close relatives would be infected the same way that we are. We share a lot in common with them, and so a lot of apes are capable of contracting HIV for that reason, right? And by the way, the specificity of those viruses, the host specificity, is another way to differentiate viruses from one another, to differentiate species or strains of viruses. So we also, that's really important, okay, to understand. Now, the specificity, the host specificity, this really emerges from evolution of the virus, the pressure that a virus is constantly under to be more and more effective. Okay? Remember that any virus, it may have many mechanisms for parasitizing, for infecting a host. But the host also has a number of defenses that it can employ. Okay? And the more specialized a virus, typically the better that virus is at overcoming host defenses. So, and that kind of thing is going to vary a lot by species. That not just species of virus, but species of host. That hosts are going to have quite a lot of variation in terms of how their immune system works and what we are able to target and how we do that. And so viruses, it helps a lot of the time to be very specific because you can make better use of the hosts that you can encounter. you're more and more likely to succeed at parasitizing one of those hosts than you are to parasitizing a host that maybe you haven't you haven't encountered before, or you rarely encounter, and you're not necessarily as well equipped to get past their defenses. So the strategies for infection, they usually involve a mix of things, avoiding those host immune system recognition defenses. So like making sure that your immune system can't see them when they get into your body. And then if your immune system does recognize them, being able to avoid being destroyed once the virus is recognized is also really important. So the more specific a virus is, typically the better equipped they are to do one or both of these things in a specific host. So this specificity, when we talk about host specificity, this actually goes beyond host species. So viruses are often very specific about what type of cell that they infect as well. So some species of virus will only infect specific types of cells and not other types of cells. Okay, so if we go back to HIV, talk about HIV. Well, HIV is really interesting because it only infects specific host cells, mostly in the immune system. Okay, so it really targets a lot of T lymphocytes and a couple others, that are mostly involved in our immune system. And so because of that, right? Because of that, this really affects the way that symptoms for these viruses develop. These viruses cannot infect other types of cells that are more common in our body, like our muscle fibers, like our osteocytes, like our neurons, anything like that. But they have a very limited range of what they can infect. Okay, the specificity that they require to do their job makes it so that those other cells, they don't even have access to them. They only have access to these specific cell types. Okay. And this is something that is seen pretty widely in other viruses. It's not a guarantee that all viruses will function the exact same way, and certainly there are some viruses that are rather general, but a lot of viruses are very specific in this way. So I like this figure. This is a pretty interesting one that kind of shows all the different things that HIV can target, right? So it's not exclusively just helper T cells, but there's, we have many types of cells in our body, and HIV really only targets a handful of those cells, right? Specifically, these macrophages. We have these like intestinal, epithelial, and lymphoid tissue. And then we have like these inflammatory cells as well, and these are the helper T cells down here that it targets, right? So all of these things, it does also impact a couple types of brain cells as well, as what we're finding, like some neurons in the brain. But by and large, it's not tackling, for example, your liver. It's not targeting your muscles, your bones, your skin, anything like that. It can't. It physically cannot. Another important aspect that we should talk about with viruses is the concept of a zoonosis. Okay? So it is sometimes the case that viral infections can spill over from one species to another. You may have a virus that is very specific, and it only targets, for example, bats. Only infects bats. Bats. And then that virus mutates, and it creates the opportunity for that virus to now maybe it's not even as useful as a bat pathogen, but it happened to have evolved a way to target humans, right? And so it is possible then for this infection, if humans are in close proximity to bats, for the virus to spill over from bats to humans. Okay, this, by the way, is the current, as far as I've last checked, the current idea about how COVID evolved. As far as I'm aware, I think the leading suspect is a bat, but it could have been a few other species as well. Coronaviruses are not entirely uncommon, and so we have some different likely suspects, I should say, for the species that it came from, the original host. And in the sort of story about how this virus spilled over, we have really good evidence to say that it did so because of the interactions that humans were having with different types of animals in these what we call wet markets, where these places where lots of animals were kept together live and sold live to people who are going to butcher them for food. So this was something that was going on a lot, still is going on a lot in different parts of China, and so this is where we believe the COVID-19 virus emerged from, right? So that is a really good example of a spillover event where viruses, as they work their way through their native hosts, they reproduce. And they reproduce and reproduce. And eventually, mutations will arise. Notably, viruses don't have as many controls over their reproduction as we do. And so they don't really have the ability to proofread their own genetics after they've been replicated. So mutations tend to be more common and happen more rapidly with viruses. That combined with their rapid production rate, where you will have new viruses being produced constantly in an infected individual. Thousands, hundreds of thousands, millions, billions at times of virus particles that are being produced. Some of those are going to be mutants. Now, of course, some of those mutants are not going to be very successful, but some of them might. Some of them really could be successful. So in those situations, when a virus mutates, it can become better at targeting a specific host, even if it is not the host that it originally evolved to target. It could, just by coincidence, evolve a mechanism that makes it better at attacking a different host. Now, if that virus never encounters that host, it is never useful to them. But in a situation where a virus does get to encounter that other host, then the opportunity is sort of unlocked, okay? And we have the ability now for that virus to work its way through a new population. This process, this is what we call a zoonotic event or a zoonosis, okay? It's that situation where a virus can switch hosts from one species to another and become a problem in that new species. Okay, and again, usually this is facilitated by close proximity between those two different species. Okay. Now, this is the sort of alternative to this, okay? You can also have viruses that make contact with an unideal host as well, where the virus may still be able to parasitize and infect that host, but it doesn't do as good of a job as that. It's not sort of the ideal host. Lots of viruses are like this, where they have a very special host. They have a host-specific target that they go after. But they have the possibility to infect other species that are maybe not as ideal of hosts. For example, influenza. is a good example of this, where there are some strains of influenza that are bird flu. Okay, um, these like H1N5s, H1N1, different things like that, or swine flu, different things like that. And there are many strains that naturally circulate out in these wild populations of birds or mammals, or in captive populations like in farms. And they are very targeted. They really only want to work on those birds like chickens, turkeys, whatever, or whatever the host is. But because humans find themselves in close proximity all the time to those infections, it's possible sometimes for mutants to jump over. A lot of the time, it is not successful when that happens. A lot of the time, when a human becomes infected by bird flu, right, by avian flu, the ability for that flu to be propagated, to be spread around from like one human to another is highly limited, because it's much harder for a human to catch that virus, especially from another human, compared to a bird catching it from another bird. Okay, that the virus just isn't equipped for that. But that doesn't mean that it's not a risk. Okay, like right now, as I record this, there are news articles about new cases of avian flu that humans are contracting, and people are very concerned that this virus may be spreading from human to human. But thankfully, at least at the moment, the evidence doesn't seem to be the case that the virus is spreading from person to person. Instead, these people tend to be people who work with birds periodically or for a job, or they're hunters, different things like that. It varies. It varies a lot. Now, of course, this is something being currently researched as I'm talking about it right now, so it's hard to say with certainty, but that has certainly been the trend throughout history, that many cases of these avian flu infections have petered out like that. Another really good example of this, though, a very notable historical example, is cowpox. Okay, where cowpox it's a viral disease that targets cattle, but humans could also get this disease. It just really didn't do much in us. It was not very, it wasn't especially comfortable or happy. You didn't feel good when you got it, but it wasn't all that life-threatening, and it also really could not spread easily from person to person. I'm not entirely sure if there are any cases of cowpox spreading from person to person. But this was something that researchers, specifically one guy, Edward Jenner, kind of realized was quite useful to us, actually, because as it happened, cowpox was very similar to a very notable human disease called smallpox. So smallpox was incredibly deadly. If I recall, I think you had like a 35% chance of mortality whenever you contracted smallpox, and smallpox would also it would maim you. It would cause such incredible like painful mutilation, basically, of the body, that it would physically scar you for the rest of your life a lot of the time. Some people got lucky and they got mild cases that were localized different parts of their body, and those places became scarred. But other people could get smallpox across the entire body, and if it didn't kill you, it left you in incredible pain, and you would walk with those scars for your entire life. So it was an incredibly destructive, horrible disease. Thankfully, it no longer exists today, as far as we're aware, outside of specific laboratories. And so we've successfully eradicated that disease thanks to vaccination. But cowpox was sort of the link in the chain to help us understand how to fight this thing. or a major link in the chain, I should say, because Edward Jenner figured out that if you could expose people to cowpox, you could make them immune to smallpox. Because they were so incredibly similar, they were actually quite closely related, and so your immune system, if it could recognize cowpox, it would also recognize smallpox, right? So that was a major advancement and discovery, and this is actually the idea of inoculating somebody with a cowpox infection. Is where we even get the term vaccination from, right? Like vaccination, the vax part of that, VACC, right? That refers to cow, right? That is what we're talking about. Okay. Anyway, let's talk about the viral life cycle a little bit. So, viruses, they're going to be pretty diverse in terms of how they do this, how they infect and who they infect. There's lots of different tactics out there, but at its simplest, all viruses must do the following things. Number one, they must insert their genetic material into a host cell. Okay, they do this. This involves bypassing the host defenses to get into the cell membrane in some way. Then, number two, they have to utilize the host cell tools to make more of itself. Okay, it's going to steal resources, hijack cell enzymes to do these different jobs, and the cell is going to be tricked into making all of the material, both the genetic material, the nucleic acid of the virus, the capsid of the virus, any spike proteins, different things like that for the virus, other features. And also along the way, avoid detection and response by the host's immune system. And finally, number three, the last step involves escaping the cell to find new hosts after that point. Okay? Oftentimes, this involves literally tearing the cell apart, breaking it apart, doing something we call lysis. Cell lysis, which means breaking apart. Now, these viral infections, okay, again, those strategies are really variable, but if we look across like large groups, we're going to see major differences. For viruses that infect animals like us, okay, there's a lot of important strategies that involve getting past the cell membrane. Which include these envelope strategies. So a lot of these like envelope viruses that will merge their envelope with our cell membrane and then take some of it with them as they leave, different stuff like that. Or they might trick our cell into eating the virus using endocytosis to just absorb the virus, to take it into its body, and so the cell enters inside of a vesicle, and then it is sent to be digested, but can't be, right? So they do get digested in a lysosome, but the parts, once they're broken apart, they don't break down into their smaller units, and so they can start doing their viral jobs inside of the cell at that point, right? There's a few different ways that might happen. In all these cases, in all of these cases, the genetic material gets its way into the cell, and the cell becomes tricked into making more of that virus. So they're going to make all the proteins. It's going to build those little capsomeres that are going to link up together to form a capsid, and then just assemble the whole virus. Its job is to make virions, okay? Remember this term, virion. A virion is a fully assembled viral particle, okay? Is a fully assembled viral particle. And so sometimes they have to escape the cell by lysis. They tear the cell apart. Sometimes, usually in the case of an enveloped virus, they will release more steadily over time, not tearing the whole cell apart, but instead stealing bits of the membrane as as the virus departs. Okay. So this is another kind of nice image of what that sort of looks like. Okay. Again, we have an example of this budding in process here. So a capsid might come in, link up with the cell, and be sort of tricked into being taken up, or maybe it just straight up inserts the genetic material right into the cell and leaves the capsid outside. Okay, this is that direct penetration. The membrane fusion is a tactic that's more involved with the envelope viruses, where the envelope of that virus merges with the cell membrane, and as it does, it just spills the contents of the envelope, which includes the capsid and the genetic material, into the cell. Okay, those are like some of the more major processes. This is the endocytosis part I was telling you about, where the cell, encounters the virus and goes, "Oh, I should eat and destroy that," and it does so, brings it inside, and then as it tries to digest the virus, that effort fails. The virus is sort of released and then breaks apart inside of the cell, releasing its genome in order to be utilized. Let's take a minute and look at RNA viruses, okay? Because some of these viruses are really interesting. Some of these RNA viruses are what we call a retrovirus, okay? Now, a retrovirus is a virus that is going to incorporate its genome into the host's genome. Now, how does this differ from a regular virus? Well, other viruses, they don't ever touch the cell's genome. Instead, they hijack some of the other tools that the cell has to transcribe and translate their genes, or to like duplicate, to replicate them over and over and over again. Okay, but they never mess with the cell's genes specifically. They never go and start inserting or removing things. Okay, but retroviruses do this. So what their job is basically is they want to insert their genome into our genome. Okay, and when they do that, then it doesn't matter whether the virus is removed, whether there are no viruses in this cell. Okay, from that point forward, the cell has the ability, and may very well do this at one time or another, just start making more of that cell. Basically, it makes it so that cell can make more of that virus continuously, right? Indefinitely. In many cases, for the rest of that cell's life, and anytime that cell replicates, it's going to pass on that piece of the viral genome. Okay? So how does retrovirus do that? Right? Well, it uses a special enzyme called reverse transcriptase. Okay? What this does, so remember that this is a virus, this is an RNA virus. Okay? It's a single-stranded RNA virus. Reverse transcriptase is going to take that single-stranded bit of RNA, and it's going to make a DNA copy of that RNA. Now, normally things don't move in reverse like that. You don't make DNA from RNA, you only make RNA from DNA. But viruses, again, they don't play by these familiar rules. They kind of do whatever they want in many cases. And so reverse transcriptase is a great example of that. We are taking an RNA template and we're making DNA from it. And then it's going to take that piece of DNA and it's going to stick it right in the middle of the host's genome in a specific place. It doesn't just insert it randomly, right? There is a degree of targeting where the enzyme has sort of like a recognition aspect, where it's very much like a restriction enzyme, has like a specific site, specific type of restriction site that it targets. And there are some really prominent examples of these retroviruses, right? A lot of these viruses are lifelong viruses, things that you get and you may have them forever. Whether or not you are having symptoms of them, you simply have the virus forever. So HIV is a very prominent example where today we can treat HIV with drugs to make it so that the symptoms of HIV are so reduced that people can live a very normal life. But they must be on those drugs for the rest of their lives. Without that, the viruses continue to be made, and it will impact their health, their immune system, and eventually, can kill somebody. Another more common example, and I suppose less feared example, is chickenpox. Chickenpox is itself a retrovirus, okay, and it's a really prominent one, okay. So, we can talk about that again in a minute, but just to cover retroviruses a little bit more, okay. Retroviruses can function very much like a latent infection, the way that they work, okay. So what does that mean? What's a latent infection? Well, a latent infection is an infection that has, you have a primary infection, and then there's a delay, and after a while, the infection can recur. It can occur over and over and over again at different intervals, and there are conditions that can cause this, right? So a retrovirus can do this because the conditions for that infection are there in you after this primary infection forever. For the rest of your life, we just live with that. Any of us who have contracted chickenpox before, not who got the vaccine, but who had the infection, we have that virus, the genetic material for that virus in us, just at all times now, okay? I speak as someone who has that, so I have a story to tell after this about that. But this is an infection that can persist, oftentimes without manifesting symptoms, and then after a little bit, a recurring infection can occur, right? So it transitions from an asymptomatic infection where you have no major symptoms to a symptomatic infection where now you're getting the viral production and it is causing your cells to suffer. It's causing symptoms to happen, right? And then you are infectious during those periods of time as well, right? You're making the virus, you're spreading it around, okay? Now, latent viruses are not exclusive to retroviruses, but nearly all retroviruses are latent infections, right? Or they can cause latent infections. This differs from other kinds of infections, like an acute infection, which is more like a case of influenza. You get influenza, you cause a primary infection, you get a big peak of symptoms and intensity, the viral production, and then after a while, your immune system kicks into gear and it cleans house, okay? Then you remove all those viruses, and now you no longer have the virus in you at all, right? There is no more infection after that point unless you lost your immunity and got reinfected, right? Or your immunity waned and you got reinfected, okay? Now, that also differs from a persistent or chronic infection, right? Where these are something where you have a primary infection, and then the infection just never goes away. Okay, and it continues at lower levels, just kind of continuously, right? So that is a difference here. A latent infection will have periods of seemingly no infection at all, where you are asymptomatic, you are totally fine, and then you can get bouts of recurrence of that infection. Okay. There's also the possibility for a slow infection, right? where you have, again, one major initial bout of infection, that primary infection, and then things go back to normal. You feel like you beat it, and then over time, you get a slow ramping of the conditions of that infection, okay? Where it gets worse and worse and worse and worse and worse. As opposed to a latent infection, this has a cumulative effect, where instead of like a baseline 0, 0, 0, 0, 0, and then a spike of a secondary infection, here you have the slow building because the virus never really stopped doing its job. The virus kept on acting. Okay, this is the case for HIV, which can turn into AIDS, right? So we have HIV, that initial infection, and when you contract HIV, you may have short-lived symptoms that then wane and you feel normal. But over time, you build up to a worsening and worsening condition until you have AIDS, right? Where your immune system really fails and you are at much higher risk of a lot of negative impacts. Okay, so this is a slow infection as opposed to a latent infection. Now, to go back to chickenpox as the story, I think is really useful for this, because latent infections from a retrovirus, the way that they emerge, it actually comes from changes to gene expression. Because consider this. You imagine that you're a kid and you contract chickenpox, like I did, right? And so you have this initial infection. It's not very fun. It's pretty miserable, and you ultimately, your parents keep you from scratching all of your itchy elevated, the pox, and eventually you get over it, right? And it feels like, oh, it's gone, right? You can go years and years and years, decades without having any more symptoms, and it feels like that was just a moment in your life, right? And then it's gone. But the virus is still there. What has happened is that your body, when you were infected, identified the source of that infection, specifically the genes that are active and producing viruses, the viral genes that were inserted and are still active. And those cells go, hey, we should not be making this stuff. It's making us sick. Let's just turn off those genes completely. So the cell can't remove genetic material once it's inserted. At least our cells cannot. So instead, we just turn off those parts of our body, those parts of our genome, right? And so we no longer make the proteins that are responsible for causing chickenpox, okay? And then we go about our lives as if everything is normal, right? And for all intents and purposes, it is. But that change to the expression of our genes is not permanent. It's not necessarily permanent. We could live our entire lives from then on without having a reoccurrence of symptoms. But if something were to happen that would cause that gene to be active again, suddenly the virus is being produced again. We are now making chickenpox virus, but at a different time in our lives and in a different way, kind of, right? The way that it sort of manifests becomes different. And this is actually how we can get shingles. So I had that experience myself as well. When I was doing my PhD, guess what? PhD is really stressful. And your body, when it is under a lot of stress, can activate and deactivate different genes in response to that. Sometimes that means accidentally activating these disease-causing genes that you contracted from a virus. And so I got shingles because when I was a kid I had chickenpox, and those genes are with me, and they were off for decades until I got really stressed out. And then my body, while trying to cope with that stress, opened up the floodgates basically turned on these genes that should not have been turned on, and then I manifested symptoms of shingles. Okay, the same thing can happen to any of us who have contracted it. Notably, anyone who got the vaccine for chickenpox as when they were younger, or an adult, it doesn't really matter. If you did not contract chickenpox and you got the vaccine, you do not have the genes for chickenpox in you, and so at that point you cannot get shingles that way, right? So that's really yet another value of vaccines, right? Vaccines are exposing us to parts of a virus without exposing us to the infectious virus itself. So our immune system becomes trained. It says, "Oh, that's that's chickenpox virus. We can target it. We can kill it." Before we ever get encounter it, before we ever encounter chickenpox at all, and so this basically makes it so that we don't have to worry about getting shingles, this much more devastating and physically painful disease that can be caused by this latent infection, right? If we nip it in the bud early, right? Okay, this whole process is, again, horizontal gene transfer in action, right? It's horizontal gene transfer from viruses to humans, okay? So even when we recover from a viral infection, it is possible for, if it is remember, it has to be a retrovirus, but in those situations, those genes that were given to us by that virus can stay with us, okay? Usually this only occurs in our somatic cells, aka our body cells, right? Like our skin tissue, our nervous tissue, our muscles, things that are not going to, reproduce to become like sperm or egg cells, right? So, in those cases, that those genes, when they're introduced to us, they stay with us. So when we have children, they don't get passed on. However, it is possible for some of these infections to occur in a way that makes our reproductive cells, our meiotic cells, get these insertions, okay? And in that situation, the result is when we reproduce, we have the possibility to pass that on to our descendants, to our children, and then ultimately to their children and to their children and so on, right? And so this is only possible for our meiotic cells, right? Our egg and sperm cells, and specifically the cells that make them, right? If this occurs in our somatic cells, well, they're just going to die with us, right? We don't pass on our muscle cells to our children. So a retrovirus that attacks our muscles is going to die with us. But in this way, if you have a virus that attacks these cells, and then you pass on the genetics that were inserted there into our next generation, right? Horizontal gene transfer can become vertical gene transfer. And so the genes that we inherited by these, pathogenic viruses can become part of our lineage. This seems rather scary, but I want to say that this has already happened throughout history. It has happened so much that we don't even think about it, okay? And throughout life, it has been a common occurrence, okay? And in fact, about 8% of our own genome that we know of is viral in origin, okay? Look at how many base pairs that is, right? 501 million base pairs of material is composed of viral compounds that our ancestors and their ancestors and their ancestors inherited. Okay, and there may even be weirdly, an evolutionary benefit to this, right? It's the introduction of new information, okay? And mutations, initially, this infectious material is bad, right? But if mutations occur on these inserted genes, we could potentially turn these bad things into really good things, right? And there very much is some evidence for this. A number of our genes that are involved in how we give birth today, right? The origin of the placenta is something that we today associate with genes that emerged from a virus, right? From several viruses, not just one virus. But and so this is essentially a way of making something good from a bad thing. It's really interesting. Okay. We're nearing the end. I want to talk about bacteriophages, which are very, very important in biology. Okay. So bacteriophages are a very abundant type of virus that only targets bacteria. It literally is incapable of infecting our cells or any other kind of plant or animal or fungus or anything. They are bacteria specialists. And they're so influential that they cause up to 70% of death for bacteria in some environments, specifically in like marine environments, like in the ocean, right? And there's still a very large portion of the mortality of viruses in terrestrial areas, like on land, right? So they're really, really important. Okay, they help keep bacterial populations in check. So across Earth, we have an estimated 1 trillion phages, 1 trillion bacteriophages for every grain of sand in the world, right? The numbers that we deal with viruses, because they are so small and there's so many, it's kind of staggering to think about, okay? So how do bacteriophages work? Well, bacteriophages are pretty interesting because they're a naked virus, right? They're familiar to us in this way. They're a naked virus, so they have a capsid but no envelope. And they also they can be either a DNA or an RNA virus, right? I think they're mostly DNA viruses, but they can also be RNA viruses. But they also have a few other important features. They have this additional complex here, which is made up of a few things. We have a sheath, a tail, a base, and these tail fibers. Okay, so this is kind of what we're looking at here, where we have these little legs. Those are the tails, right? The base is like this bottom part that meets up with the cell when it attaches to it. The sheath and tail. I'm sorry. I meant to say that this is the tail, and then these little fibers that come off the little legs. So the sheath and tail are that mechanism that essentially allows the bacteriophage to penetrate the bacterium and insert its genetic information into it. Okay. So these bacteriophages, they replicate using two methods, and they alternate between these methods. Okay? So the first one to talk about is called the lytic cycle, or the lytic cycle. Okay? It's lytic specifically because it is lysis, it causes lysis, right? So the lytic cycle is something that, it's a type of replication that makes the replication more immediate, right? It happens like right away. Okay, so how does this work? Well, the viral particle or the virion attaches to the cell, and using that this structure right here, right, it inserts its genetic information into the cell. Okay, so the genome enters the cell. The bacterial cell is tricked into transcribing and translating the materials into protein. It also will make more of the genome, right? So it replicates everything. It replicates and makes everything. And then inside the cell, those viral parts, they become assembled into new virions. Once those virions are assembled, they build up to such an abundance that the cell tears itself apart, spilling those viral particles into the environment so that they can go and infect new cells, new bacterial cells, right? So the result of this, method, the Lytic cycle, is the rapid replication of the virus, the quick replication of it, and the quick death of that host cell, right? Like the host cell is going to break apart and release a lot of those materials. Once the cell is broken apart, there's no saving it. It's dead. Then, separate from the Lytic cycle, we have the lysogenic cycle. Okay? This is more of a long-term replication strategy, where a bacteriophage, it starts the exact same way. Okay? A bacteriophage attaches and inserts its genome into the host cell. Okay? But instead of starting the Lytic cycle, the genome is going to integrate itself with the host genome, very much like a retrovirus does, right? Very much like we talked about with a retrovirus. Similar, but not necessarily the same mechanism. Okay. Now, from that point, that bacterial cell, is going to go about its daily business. It's going to grow. It's going to replicate just like normal, not expressing any genes from that virus. And it does this because the genes are silenced at this point. They are turned off. Again, very similar to the way that retroviruses work with us. So the cell is going to keep on doing its business. It's going to divide and divide again and divide again. Its offspring are going to keep on dividing. And it's just going to behave like totally normal, like it doesn't have a time bomb sitting inside of it. Once activated, though, right, eventually that cell is going to have those genes turn back on for one reason or another, usually when the cell becomes stressed in some way. You know, again, very similar story. Once the cell activates those genes again, those genes become productive. They are being made. So the particles, the parts that make up the virus are made. New virions are assembled, and the Lytic cycle begins. Right? Now the cell bloats, it expands as the contents are expanding, as all these virions are being packed inside of it, and eventually the virus tears itself apart and dies. So in this way, we have a cycle. We have a circular type of event where a cell may be infected with a virus, and it might just do the Lytic cycle, or it might start with a lysogenic cycle, in which case the cell is replicating, going about its business like everything is normal, and then eventually something triggers the Lytic cycle to begin with that virus, right? With that bacterial cell and the virus inside of it. So this is a nice explanation of this, where here's the Lytic cycle up here. We've got the bacteriophage inserts this material into the host cell. The phage DNA and the host DNA are separate at this point, right? And then the cell is tricked into making more of the viral material, but it makes so much the cell eventually breaks apart, releasing the bacteriophages. Or the lysogenic cycle can start, where at the same point the cell inserts its information. That genetic information instead becomes part of that original host. Okay, the host continues on. It divides, doing its own thing, right? And then eventually, stressful conditions cause the cell to change the way that those genes are expressed, and it starts causing the the virus to leave the lysogenic cycle and enter into the lytic cycle, right? Entering in at this point right here. Okay. Now, don't forget as well, this links up with our understanding of bacterial transduction. Okay, so this process happens normally inside of viral infected bacteria, but transduction happens when this process, that what the virus is doing, does not go according to plan, where something gets a little bit messed up, and the viral DNA gets inserted, and it doesn't, maybe the cell can't get into the lytic cycle from the lysogenic cycle, like a mutation emerges and it messes that up. Or when the cell switches into the lytic cycle, you get a different amount of information that is a different piece of DNA that is cut out and used to build the viral DNA instead, right? So this virus, for example, in this case, has somehow gotten a hold of bacterial DNA instead of viral DNA, right? So if it goes to insert itself into a new bacterium, it's now going to insert bacterial DNA instead of viral DNA, which will not have the same kind of viral lytic lysogenic relationship. It's not going to do that process, but it may still insert into the bacterial genome, right? This is how a gene that is useful in one bacterium could make its way into another bacterium. Could just as easily happen with genes that are not useful too. Okay, so this is another example of horizontal gene transfer between bacteria, but this time using viruses as an avenue to do that. Okay, so that sums this up. Please make sure that you review the contents of the chapter really well for this, because there's a lot in this chapter. It's kind of complicated content overall, because we've talked so much about bacteria at this point, not so much about these infectious particles. Make sure that you know the life cycle of these viruses. About retroviruses, about bacteriophages, about these lytic and lysogenic cycles, that you can categorize these bacteria, and make sure that you also go and review prions and viroids as well. Those are in this chapter, and also in previous ones. We have spent a lot of time talking about prions in a previous chapter, so make sure that you thoroughly review them. Okay. Thank you, and I'll see you next week.